Chapter 3, Section 13, Sugars and Polysaccharides. Now, polysaccharide biosynthesis is going to be tied very closely to gluconeogenesis. Okay, polysaccharides, NAGs and NAMs, there's going to be a lot of them that are going to be required for the making of peptidoglycan. Or if there's a slime layer, a capsule, remember those are polysaccharide driven also, proteins and polysaccharides. So going to need those also. Uridine diphosphoglucose, UDPG, is a glucose derivative. Okay, it's going to be the precursor for our NAG and NAM and for the sugars attached to the lipopolysaccharides. You're also going to see adenosine diphosphoglucose, and then any glucose that's going to be added to, form polymers. So that whole idea of gluconeogenesis is the making of glucose, sugar making, glucose, sugar, neogenesis is the making of. It's the starting off with something that is not a 6-carbon sugar and building it back up. They're literally reversing glycolysis to an extent. Taking pyruvates, taking acetate, taking other things, oxaloacetate, and working in reverse to build things back up, to reverse the catabolic reactions of glycolysis. The uridine diphosphoglucose, UDPG. UDPG. Here's our ribose, here's our uracil, 1,2-phosphate, and a glucose. By targeting glucose this way, it sequesters it away from glycolysis. It means it's now going to be readily available to enzymes to start forming the basis of NAG and NAM. This whole idea of gluconeogenesis is the taking of, as I said, 2-carbon, 3, 4, 5-carbon strands that could be fed into the citric acid cycle, consumed that way, or it could be components of the citric acid cycle, hence the oxaloacetate. And you reverse the process. Oxaloacetate to phosphoenolpyruvate. Hey, remember phosphoenolpyruvate? That's the step right before, pyruvate, the end of glycolysis. Well, you take phosphoenolpyruvate and you work backwards. And there you go, ladies and gentlemen. You now have G6P, glucose 6-phosphate. Glucose 6-phosphate that can now be tagged to become UDPG or adenosine diphosphoglucose. Or the G6P can be utilized in what's called the pentose phosphate pathway. Pentose phosphate pathway is a mechanism for taking glucose, a 6-carbon, 6-sided ring, and turning it into a 5-carbon, 5-sided ring. In the process releasing CO2. Why would we need that? Well, that's, isn't a 5-carbon ring, blah, 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 fructose? No, fructose is a 6-carbon, 5-sided ring. Looky, looky. 1, 2, 3, 4, 5. 5-carbon, 5-sided ring. Ribose. G6P gets turned into ribulose 5-phosphate, releasing CO2. Ribulose 5-phosphate becomes ribose 5-phosphate, which becomes RNA or DNA nucleotides. So this pentose metabolism, pentose phosphate pathway, is a means for making 5-carbon rings, C5 sugars. Basically, ultimately you want to end up with ribose. You could start with glucose or hexose, remove one carbon atom, boom. Or you can take other carbon strands and in the gluconeogenesis, build up till you get G6P, glucose 6-phosphate, which you then go cut, remove that carbon dioxide, and you end up with C5. So this pentose phosphate pathway is the major pathway you see for the making of the sugars that are going to be utilized for the making of nucleic acids. Glucose oxidized to, CO2, NADPH, ribulose 5-phosphate. Ribulose 5-phosphate is not the correct one. Okay, ribulose 5-phosphate will then be twist, tweak, cut, pasted, and all of that to form ribose. The majority of the pentoses will be catalyzed through this method. By reversing the method and going through alternate mechanisms to get to this point, you can have carbon strands of multiple different lengths. 4 carbon, 5, 6, 7 carbon strands. Glucose 6-phosphate, once it's made, gets oxidized, 6-phosphogluconate, and then one of the carbons gets cut off, and it further gets oxidized until you now have 1, 2, 3, 4, 5 carbon ribulose 5-phosphate. Ribulose 5-phosphate can then be turned into ribose, which can then be used nucleic acids. Now think about it. Some of the things you ate, okay, whether they're plant or animal based, you're going to be eating cells. In those cells will be nucleic acids. You're not going to directly absorb the DNA of things. Okay, I'm sorry, those people who think you're going to directly absorb DNA strands from genetically modified organisms. That's not how it works. You're going to break down the chromosomes into individual nucleotides. Those nucleotides get broken down, consumed, and you're going to have ribose 5-phosphate, C5. You take a couple of these and you link them together. You get a 10 carbon thing that you can then start breaking up. Here's a 7 carbon and a 3 carbon, still 10 total. You then rearrange, swap things around. You end up with a 6 carbon strand, which you then kick over to that glucose making, gluconeogenesis. The resulting 4s, you bind to something else, another 5 carbon. So 4 and a 5 makes 9. Split that in half. You now have a 6 carbon thing and a 3 carbon thing. That could be, pyruvate. That could be another, hexose, glucose 6-phosphate, or whatever. And you just keep going and going and going.